Hello friends, in this module we will look at the metalloenzyme superoxide dismutase. Now we know that enzymes are the catalyst of biological systems. They not only control the rate of the reactions but by favoring certain geometries in the transition state can lower the activation energy for the formation of one product rather than the other. The basic structure of enzymes is built of proteins. Now, metalloenzymes are composed of a protein structure called an apoenzyme and a small prosthetic group which may be either a simple metal ion or a complex metal ion. A reversible bound group that combines with an enzyme for a particular reaction and then is released to combine with another is termed a coenzyme. Both prosthetic groups and coenzymes are sometimes called cofactors. Now we have a number of enzymes under different classes which are present in some of the biological systems. For example, we have hydrolases. Examples include carboxypeptidase, carbonic anhydrase, etc. Then there are oxidoreductases, which include superoxide dismutases, cytochrome C oxidase, nitrogenase, etc. Then there are isomerases and synthetases like vitamin B12 coenzyme, and also there are transferases, for example, the kinases. Let us look at the enzyme which contains copper and zinc, which is called a superoxide dismutase. Now we know that cells rely on oxygen as the final acceptor of electrons in respiration, allowing us to extract far more energy from food than would be possible without oxygen. But oxygen is also a dangerous compound. Reactive forms of oxygen such as superoxide, they can leak from the respiratory enzymes and wreak havoc on the cell. This superoxide can then cause mutations in DNA or attack enzymes that make amino acids and other essential molecules. To combat this potential danger, most cells make superoxide dismutase, an enzyme that detoxifies superoxide. So the superoxide dismutases are enzymes that catalyze the dismutation of superoxide into oxygen and hydrogen peroxide. Thus, they are important antioxidant defense in nearly all cells exposed to oxygen. There are three types of superoxide dismutases that are known. They are characterized by having copper, manganese, or iron as cofactors. The copper protein has zinc plus 2 as an essential component. Thus, the three types have been named as copper zinc superoxide dismutase, Mn superoxide dismutase, and the iron superoxide dismutase. We will look at the copper zinc superoxide dismutase in detail. The copper zinc superoxide dismutase molecule has a blue-green color suggesting the presence of copper plus 2 ion. The molecule has a cylindrical shaped structure and consists of two equal subunits. Each subunit has one copper plus 2 and one Zn plus 2 ion. The copper plus 2 ion is coordinated to 4 nitrogen atoms of the imidazole from histidine residues. A water molecule occupies the fifth position, resulting in a square pyramidal structure. As you can see in the active site of the copper zinc superoxide dismutase, the zinc ion is bound to 3 nitrogen of the histidine residues of the protein, and the fourth position is occupied by an oxygen atom from the aspartate group, giving an approximately tetrahedral geometry. The copper and zinc of a subunit are breached by a deprotonated imidazolate of histidine and are at a distance of 6.3 angstrom unit. If copper and Zn plus 2 ion are removed from the enzyme, the apoprotein gets deactivated, but the activity is revived on addition of copper plus 2 only. This shows that copper is the active site of the enzyme. Zn ion has predominantly structural role in the enzyme. Zn has all the four coordination positions occupied and does not have any accessible coordination site. Hence, it cannot participate in the enzymatic process. The superoxide binds to copper by displacing one of the histidine groups. So the main role of the superoxide dismutase enzyme is to dismute superoxide. Let us see what we mean by this term dismutation. Dismutation is a term that refers to